So based on my title, I took a different approach and decided that I would spend most of my time just educating you about hepatic transporters and giving you examples of how transporters can perhaps be involved with toxicity, altered disposition, and then I'm going to spend a small portion of my talk kind of discussing some data that we have with bisphenol A. So before I begin, I just have to say that I wistfully sit back sometimes and listen to P450 and phase 2 lectures and think like, oh, I wish I should channel that in my lab again. Because when you are working with transporters and you go from doing expression work to actually categorize, understanding the function, there's a lot of technical challenges. So that kind of presents a challenge to us in the field of going from whole liver to being able to understand activity. Okay, so today we're just going to go over the major hepatobiliary transporters and their impact on drug disposition and toxicity. And then I'll talk about some EHS-related work with bisphenol A, in particular looking at transport of BPA glucuronide and also a developmental model of BPA. Okay, so this is going back to like Pharmacokinetics 101. So we know that as far as total drug clearance, that the liver is one of the components to total drug clearance, and hepatic clearance quantifies the loss of drug during its passage through the liver. With regard to the components for hepatic clearance, we'll consider metabolism as well as biliary clearance, and just to appreciate that transporters are going to sometimes impact and contribute to hepatic clearance mechanisms. So with regard to the field, you'll hear about the extended clearance concept, and these are kind of models that are coming out that are integrating transporters into the kind of pharmacokinetic predictions. This is a very complex slide that's showing the interplay between transporters and P450s for bile acid recirculation. So first of all, just to appreciate that transporters just don't only exist in the liver, although that's what we'll talk about today, but they also exist in the intestines and the kidneys. In this slide, this is focusing on how transporters are working in combination with P450s and phase 2 enzymes to contribute to bile acid uptake, metabolism, and then excretion into the bile, where they can undergo recirculation, utilize intestinal transports in the enterocytes, and then get delivered back to the liver again. Alternatively, it's what's called a sinusoidal or alternative export. So there are transporters that are localized also on the basolateral membrane, such as the ROSTs and MRP3 and 4, and my suspicion is maybe Jose or Nathan will talk about those, and that these can contribute to efflux back to the blood through the sinusoidal membrane, and then contribute to urinary excretion. And again, there's also transporters within the kidney that are going to contribute to the disposition of drugs. And this would be drugs as well as xenobiotics, chemicals of environmental concern. So there's generally, I wanted to go over the nomenclature for transporters a little bit. So there's basically two different classes of transporters that exist in the liver. We have our SLCs. Predominantly, these are uptake transporters within the liver, except for the mates. So the gene names here are SLCs, and then they encode for various proteins. Today in particular, we'll spend some time talking about OATPs, a little bit for NTCP and OCTs. On the efflux, on the apical membrane of the hepatocyte, our multi-drug resistance proteins, or our ATP binding cassette transporters, are going to be our efflux transporters. And these are, say, the ABCs, so for example, like MDR and P-glycoprotein, which I'm sure everybody in the audience has heard of, BSEP, as well as our multi-drug resistance associated proteins and breast cancer resistance proteins. So basically, two classes of transporters, some bringing chemicals into the liver, some bringing, pumping them out. So when we look at the hepatocyte, and I just want to take a minute to orient to you the different transporters, names, and functions. So we have our OATPs. These transport a diverse substrates, and OATPs are localized to the basolateral membrane of the hepatocyte and mediate uptake from the sinusoidal blood into the hepatocyte. We also have NTCP, which is the sodium taurocholate co-transporting polypeptide, and its major job is to transport bile acids from the blood into the hepatocyte. It should be noted that NTCP actually can transport some chemicals of environmental interest, such as perfluorinated PFOA. That's not very common to this particular transporter. OCT1, we'll use that example today. It's the organic cation transporter and transports metformin. Not on this schematic, but it would be along the sinusoidal membrane. BSEP, NRP2, BCRP, and MATES are all localized to the biocanalicular membrane. BSEP is going to be the major workhorse for excreting bile acids into the bile. MRP2, also referred to as ABCC2, so just be careful, I may exchange these back and forth during the talk. This is the newer nomenclature. MRP2 is kind of the major workhorse within the liver to get xenobiotics and drug conjugates out of the liver and into the bile. And lastly, BCRP, or breast cancer resistance protein, is actually more closely related to the multi-drug resistance associated proteins, and this is going to transport sulfonated conjugates into the bile, also localized to the biocanaliculus. This is just a different schematic basically showing the same thing as before. I just want to point out that there are species differences in OATPs. So for example, this schematic is showing rat transporters. So rat OATP 1A1 and 1A4 are more closely related to human OA1A2, and rat, or say a preclinical species, OATP 1B2, is more closely related to human OATP 1B1 and 1B3. And so these are not necessarily from the same gene products. They do share kind of functional substrate overlap. This is just a nice picture from Lauren Alex Sunis, who I wish was here today, showing showing some nice NTCP staining along the basolateral membrane, and then here's BCRP staining in the biocanaliculus. So as Dr. Proud talked about, that nuclear receptors can modulate P450 expression. Nuclear receptors can also modulate drug transporter expression. This schematic kind of shows how PXR and AHR, CAR, PPAR alpha, and FXR regulate those various transporters that I just mentioned. And what I really like about this particular slide that was shared with me is that you can pretty much see how there's similarities across the groups for regulation. So for example, most of the nuclear receptors are, say, decreasing OATP expression and increasing, say, MRP expression. Now what's not on here is some people would probably want to have NRF2 on this schematic too, and that's because that pathway is also involved in modulating transporter expression as well. So let's go over just a few of these in detail. So I'll talk to you about human OATP 1B1 and 1B3. As we mentioned, they're localized to the sinusoidal membrane of the hepatocyte. Interestingly, these can be targeted to the liver. So when I talk to my friends in industry, sometimes when they're trying to get drugs to the liver, they're actually trying to target an OATP mechanism, trying to utilize physiochemical properties to get that to go through OATP. Kurt Clausen would always say that you have to get the drug into the liver for the liver to do something to it. And so OATPs are known to be rate-limiting determinants for P450 metabolism, as well as hepatobiliary excretion of a chemical. So if you don't get that chemical in, it is also going to impact what gets into the bile. And as I mentioned, species differences do exist. And I just want to point out from an EHS standpoint that it's really important that we understand species differences between OATP and OATP substrates for human versus rat. So one of the limitations to the literature when you're evaluating environmental chemicals for risk is you tend to not find species studies that are comparing the affinities for OATPs. This is an example of OATP double knockouts. Schinkel's group out of the Netherlands made an OATP double knockout, so these are going to lack both of the predominant OATPs on the basolateral membrane. This shows that plasma methotrexate levels are increased in the knockouts. Alternatively, when you look at the levels in those knockouts, there's no methotrexate accumulation in the liver. So this was a very nice example showing that OATPs contribute to methotrexate accumulation in the liver, at least in mouse. They went on to publish a second study that was really clever, in which they then introduced, they humanized these mice with various OATPs. So here, this is a stain for OATP1B3, and showing that these humanized mice now express OATP1B3 kind of in the localization that they're supposed to. When they either had human 1B1, 1B3, or 1A2, they were able to increase the amount of liver methotrexate that was getting into the liver. Very clever. Furthermore, when we look at just not drugs, but endogenous chemicals such as bilirubin, our OATPs are very important for bilirubin. I like this slide for a few reasons. First of all, it shows very clearly that when you disrupt or knock out our OATPs, you're going to increase plasma bilirubin. But if you look, as I mentioned, you have to get something in to get it out. So in this case, not only is plasma really increased, but what has gotten into the bile decreased. So you knock out something that's at the sinusoidal membrane, it's affecting what's getting into the bile. Moreover, to prove the point, urine bilirubin excretion is up because the blood levels are higher. Okay, so how does this apply to humans? Really, one of the best examples are with statins. And this was a really great paper that was in the New England Journal of Medicine, where they looked at a large cohort of people and looked at identified certain polymorphisms in OATP1B1 and risk for statin myopathy over a 5-year period. And really what they found was that compared to having a T, TT, if you had a C, you had an increased odd for developing myopathy over time. So this is looking at cumulative percentage of patients who have had a myopathy. And so you can see that the patients that are homozygous for the CC genotype are at a much higher risk for statin-induced myopathy. This was simvastatin. Now, if you go and look through various studies where they just not have looked at simvastatin, but other statins, you'll see that other statin AUC levels are also increased with an OATP polymorphism, with the hemizygotes being somewhat increased, and then if you are a homozygous at that site, that you'll have an even more increased statin level. Lastly, Kirk Clausen's group showed a nice body of work showing that if you generate OATP knockouts, you can protect the liver from hepatotoxicity. So they administered phalloidin. Phalloidin was toxic to the wild-type mice. However, in mice that lack OATPs, they don't have the same elevation in serum ALT, and this is just the pathology showing it. So this is the knockout. If you can't get it in, it can't be toxic. Okay, moving on to OCT1. This is also a basolateral transporter. This is a nice study in mice by Kathleen Giacomini's group, showing that when you knock out OCT1, you notice metformin levels don't really change in the blood. However, when you look at metformin accumulation in the liver, it's dramatically lower. So this is a very useful study because it's showing that, if you were to just go by, if you were to assess that tissue exposure would be the same as blood exposure, you would really miss the effect. And just to also note that in this study, they also showed that there were some SNPs in human SNPs that resulted in reduced uptake of metformin. Okay, lastly, MRP2. MRP2 is mainly expressed on the apical membrane of hepatocytes, so this is staining in sandwich cultured hepatocytes and showing accumulation of a dye that utilizes MRP2 as for transport, and showing that there's a nice kind of cannulicular accumulation of that MRP2 substrate. It is the significant contributor to biliary excretion for many compounds, often xenobiotics that are forming either glucuronide and sometimes sulfate conjugates. Absence of MRP2 function can result in Dubin-Johnson syndrome in humans. This is a very clear example in rats that lack MRP2. They have an increased color of bilirubin in their serum. They're hyperbilirubinemia. Thank you for the slide. Okay, and furthermore, if you knock out MRP2, you're going to decrease what you're going to get into the bile. So these are wild-type mice that were administered, the livers were perfused with fexofenadine, and you can see that there's an increase in fexofenadine in bile over time. When we look at knockouts, there's no longer an increase in fexofenadine into the bile. Okay, so lastly, we have BSEP. This is our bile acid transporter that sits on the canalicular membrane and contributes to bile acid excretion to bile. Importantly, as far as drugs and xenobiotics are concerned, inhibition of BSEP can result in intrahepatic cholestasis. So from a drug development standpoint, you want to minimize interactions to inhibit BSEP. And this is just an example of species differences in how you have to understand whether there's perhaps a BSEP mechanism or not. So in rats, bosentan is not hepatotoxic, and that's because actually it blocks NTCP, and bile acids can knock it into the liver. Whereas in humans, bosentan does not inhibit NTCP, gets into the hepatocytes, but it does inhibit BSEP. And when it inhibits BSEP, you get an accumulation of bile acids in the bile, and then toxicity. Furthermore, David Wong's group had shown that if you make a BSEP knockout in conjunction with an MDR knockout, that you can really have profound effects for certain endogenous compounds. So for example, this triple knockout has very, very high levels of plasma bilirubin. But if you were to look at bile acid, either bile flow rates or bile acid output, MDR doesn't really contribute to the bile acid output, but BSEP is really the main contributor. Okay, so furthermore, if you can look at the physiochemical properties of molecules, you can kind of predict which route they'll go. And so there's some people at Pfizer who do this, and this is a nice paper from by Varma, where he looked at physiochemical properties and predicted what was it about those chemicals that would predict whether or not they would undergo biliary excretion. So in the classes of compounds he looked at, 60% of them would undergo rat biliary excretion. And then when they correlated those, they found that these compounds were also either rat 1b2 substrates or substrates for human OATPs, and that these compounds were, in general, more acidic. And furthermore, they looked at other physiochemical properties and just were able to kind of make some generalizations. One of them being that typically acids will undergo biliary excretion in rats. PK, so the more acidic the molecule is, as you increase the pKa, biliary excretion goes down. And there's other parameters as well. Okay, so how do transporters contribute to chemicals of environmental exposure? So our laboratory was interested in determining whether or not human and rat ABCCs could transport the metabolite of bisphenol A, the plastics contaminant of BPA glucuronide. So this is data from an ATP assay. So MRPs require ATP in order to transport. They will liberate ATP if there's transport. You can utilize the dye CDCF and measure its inhibition into vesicles. So here we see that BPA itself does not really interfere with CDCF transport. BPA sulfate inhibits it a little bit, but BPA glucuronide is able to inhibit that transport. When we look at human, a very similar pattern with BPA glucuronide being the best inhibitor for this transporter. Overall, rat had a higher Km than human and is a higher affinity. This is a little bit different than another study that's in the literature, and this is likely due to the quality of the vesicles. We ended up getting vesicles from the same vendor, and the protein expression was very poor. We also looked to see whether or not BPA glucuronide would be a substrate for MRP3. Just to give you some perspective, I just don't have more time, but in rodents, BPA glucuronide typically undergoes biliary excretion. Whereas for humans excrete the majority of BPA glucuronide into urine. And again, this has to do with the vectorial excretion. So MRP2 going to bile, MRP3 going back to blood and then on to urine. So this graph on the left shows that with increasing BPA concentration, you have release of phosphate from your ATP assay, indicating that BPA glucuronide is a substrate for human MRP3. And we can also show that we can compete for CDCF transport with BPA glucuronide. So very quickly, this is the last bit, only a couple slides. We have experimental data where we looked at developmental BPA exposure. So this is a very interesting paradigm. Moms either received corn oil, 50 micrograms of BPA per kilogram diet, 50 milligrams of BPA per kilogram diet, 0.1 microgram of phenylestradiol per kilogram diet, or a combination of 50 milligrams BPA per kilogram diet, and within combination of Geneseed, kind of utilize this as a reversal. So mom received diet. She then was put with a mating pair and continuously exposed to the BPA or phenylestradiol, had pups. Pups stayed with mom in the diet until weaning, and then the pups were weaned and they never saw BPA or EE again, and then they were evaluated at postnatal day 135. And this was done in conjunction with Cheryl Rosenfeld at the University of Missouri. So interestingly, when we assessed these mice, they didn't really have any overt liver phenotype, but we decided that we would assess their hepatic clearance. And we used a chemical called BSEP. It's actually hard to find it, but it's a very useful chemical because it doesn't undergo hepatic metabolism. It just utilizes transporters to get to the bile. So DBSP is a substrate for MRP2, also called ABCC2. When we looked at these mice and administered DBSP, we saw that in the pups from mothers that received this BPA treatment, they had a decreased DBSP disposition in bile at both concentrations. When we further followed up and looked at MRP2 protein expression and mRNA expression, BPA very selectively decreased MRP2 expression in the liver. We actually did this. It was such a hard study to believe. We actually ended up analyzing 2 cohorts of mice that were born about a year apart and saw similar results. So even though this blot's a little bit uneven, for the most part, we see that there's an explanation why, but it's too long to get into right now. Overall, BPA, both concentrations will decrease ABCC2. Phenylestradiol totally abolished it. But what was very interesting to us was that BPA, in combination with Geneseed to mom, did not cause this downregulation. So we're following up on that. That's just a little bit of information about what we're doing related to an EHS mission. I'm not concluding, but I wanted to kind of throw about, well, what's the gaps in knowledge where EHS could fill. First of all, the contribution of transporters to the excretion of chemicals of environmental concern. Linda Birnbaum's done a lot of work looking at the biliary excretion of chemicals, for example, but then that's kind of where our knowledge stops, meaning that we know that a chemical we know its disposition, but we don't necessarily know the mechanism compared to rat or human that leads to that disposition. Leading into that, species differences in transport that are resulting in hepatic effects and accumulation. A great example of this is perfluorinated compounds. They accumulate in liver. It's really important to know whether or not that species difference is partially related to OATP affinity for those compounds. And lastly, maybe people would argue with me about this one, but Les Bennett's done a very nice job of categorizing about 900 different pharmaceuticals and trying to predict, based on their physiochemical properties, where those chemicals, whether or not they're going to go into bile or urine. And this exercise hasn't been performed for environmental chemicals, but it seems like it could be a useful tool for us to utilize before we go ahead and just start plugging them into animals. So with that, there's a special group of people. Some of them are here today who have, maybe I'm not really showing data, but these are people that have made studying transporters really fun. And then we have our adjuncts at the University of Rhode Island, Eamon Yurong and Menthana, who are adjuncts at the University of Rhode Island with me. If you want more, this 3 cast of characters also happens to run a workshop at the University of Rhode Island that's heavy industry-based. It's a lot of SimSib, PK type of modeling, drug-drug interactions, FDA guidance, things that it's like hard for me to understand. And with that, I just would like to thank you for your attention, thank Matt, and answer any questions.
Questions for Andy?
Yeah.
Oh, sorry.
Yeah. Can you speculate what's the mechanism why BPA goes the downregulation of some of the transporters?
Yeah, so I didn't show it today, but we've characterized those livers quite a bit. So we do know that there's, I think there's some kind of panomic changes in transcription factor expression and binding that's happening with those livers. So for example, we know that some of the transcription factors that are involved in regulating MRP2 expression, they're just not their expression, but their binding is decreased. And then also at the MRP2 promoter, there's a decreased recruitment of histone deacetylases. Excuse me, I misspoke. There's an increased recruitment of histone deacetylases to that site in the promoter that is known to drive MRP2 expression, as well as perhaps NRF2, a decrease in NRF2 binding. So we do have a deeper mechanism. How genistein is playing into that is not quite clear, but we do know that genistein doesn't interfere with the absorption of BPA, so it's not as though mom's eating genistein and genistein is preventing BPA getting into her. It's a different mechanism. It must, it's maybe at the liver level.
Is ATP availability ever limiting in terms of the transporter function?
I can't readily think of an example, but I mean, maybe there could be. I don't know. I mean,
I guess it depends on how compromised the cell is. Is that what you're referring
yeah,
to?
I guess I'm thinking, maybe as far as like if you were thinking about a very, very damaged liver or a fasted liver or a liver that maybe
Well,
really
that's the question, how
depleted
damaged
ATP.
or fasted does it have to be?
I can't think of any study that's addressed that question, and I don't know, but I guess in theory maybe it could, or maybe there's compounds that are so perhaps. I don't think anyone's done
Well,
that.
Bob, you can have a significantly stressed cell, and it will respond by increasing the expression of some transporters. And that would, of course, require, ATP as an attempt to rescue the cell somehow, So
Right.
there might be shuttling of energy in the cell to
So if you have one
engage.
transporter, but you don't have the
Right.
ATP to shuttle,
Right. So
Correct.
pump, then
There's no example to point that out. And I would say that we tried this actually with fasted rats. And actually you're trying to perform bioaccumulations on a fasted rat. That's like an Iacoc's nightmare. And, and on top of it, actually physically trying to do it, and, not successful. So I think that the limitation is for the field is that to answer that question, it's very challenging.
So we actually did ATP dependence of the sodium pump in early 1980s, and it's quite remarkable that the Km is very high. If there's any, at least for that system, if there's any drop in the ATP, you see it as far as that activity.
we don't know whether,
Yeah.
is anything known about the KMs for ATP for any of the pumps in the liver?
It's got it.
I mean, I'm sure, but
That
I
has
guess
to be
I don't
sorted.
really, I have to really think about papers that I read about
That's
10
kind of
years
a
ago.
rough term.
Before you were
But-
born.
Most of these transporters are so most of these transporters are obviously characterized in vitro systems, and it's in vitro systems where they're purposefully limiting ATP to show that it's ATP dependent, or they're obviously adding an excess of ATP to look at substrate. So I don't think anyone actually has like KM values that I can think of offhand for the specific transporters, but of course it can be limiting it, and that's one of the ways that you show that it's an ATP dependent process.
So to stay on schedule, I think we should complete that. Thank you. Nice presentation.